All right, this mini lecture is about protein structure. And all proteins are put together the same way. And we're going to introduce aquaporin. Keep talking about aquaporin is one example. And it's a good example to pick on first, and we're going to talk about it. Now, here's where we're at, is we're building a cell, building up to chapter 6, the cell, and we're building it based on some features, and that is that all 6 cells exist in a water environment, and that introduces chemistry. What's significant about that statement is that water has a certain chemistry based on these polar bonds of water, and other molecules have polar bonds as well. Water becomes a hydrophilic polar environment, and we can have hydrophobic and hydrophilic characteristics that will affect the chemistry of life in water. And a good example of that is the next thing, that all cells are surrounded by a cell membrane. The cell membrane has has phospholipids that have both a hydrophilic and a hydrophobic characteristic. They're said to be amphipathic. That one molecule has 2 chemical characteristics. And as you know, biological molecules are big molecules, so they all have that quality where they're within a big molecule that have different regions of polar and nonpolar covalent bonds, making its chemistry. And all cells have similar chemistry. So from bacteria to people, lots of the stories are really, really similar, and they feature a lot of proteins. And so what we're doing is we are introducing proteins, and our book does as well, in several chapters, but this is mostly going to be talking about chapter 4 and 5, like that. Okay, and we're working on this part of our course. And the next thing is to remember this idea that we're always talking about evolution. So we started chapter 1 talking about big themes in the book, and one of the 7 themes was evolution, and we say, oh no, it's not really a theme, it's all pervasive, it's a pattern. And so keep that in mind, that anytime we're talking about this stuff, the similarity of proteins in all life, the cell membranes and so forth, that's a pattern, the pattern of nature. The pattern of nature is very consistent with evolution as being a process that has occurred and is occurring on our planet, and creating a pattern. Then the next thing is we want to keep track of the size. So here we're going to be talking about building a protein, building it based on these atoms, and building something that in the Boston metropolitan area would be the size of a truck. Different proteins are different sizes, so you see it's showing a range there. All these are smaller than visible by electron microscope. The electron microscope should really cut out here. This is ridiculous. It's not going to get down to that scale, as far as I know. And yeah, it just doesn't make it that far. So individual proteins are invisible to the most powerful microscopes, but there they are. They're physical, and some are the size of a big, long 18-wheeler, and some the size of delivery truck. So there's some range and sizes of different proteins like that. And we're looking at aquaporin, and we can talk about aquaporin just like a truck. It's a physical thing. It's floating in the cell membrane, and it has this overall shape, two-dimensional shape, that makes it look like a donut. And so you'll see that a lot in biology, where the protein are drawn as like a blob with a shape, like this, where that's somehow an important part of the protein. It's a blob with a shape to it. And in this case, if we dive within the blob, the shape, that donut shape, is based on a long chain of cooked spaghetti, a long chain of amino acids joined together by covalent bonds, and there's a series of spiraling shapes that are this long single chain of cooked spaghetti that create this barrel-like structure through the aquaporin. So that here's a picture of aquaporin, and you can follow this single strand of the amino acids through to make this protein. So it's a donut-shaped thing, but you can look in more detail on it. It's like a series of spirals in kind of a barrel or cylinder shape. And then the next thing is that all proteins are based on chaining together individual amino acids to join together by a covalent bond that's called a peptide bond. It's just a special name given for it's just a kind of a common covalent bond used to join together amino acids. And so here you go. Here's aquaporin. It's a long chain. It looks like it's about 150 amino acids. These are single letter abbreviations. There are 20 amino acids. And remember this idea from handout 1 that we used insulin in that example. And it is just another protein. And we have several thousands of different proteins that we make based on DNA, and we inherit from our parents. And so here's something from our book where the structure of proteins is described at these 4 levels. And you want to learn these terms. The primary amino acid sequence is just the chain of amino acids that are joined together in that cooked spaghetti. And we've got different proteins, insulin, aquaporin. They differ because they have a different pattern of the 20 amino acids. So 20 common amino acids, and they're joined together in a different pattern. Protein's a couple hundred amino acids long. Imagine a word with 200 different letters each. You know, the 26 letters are alphabet. You just add the letters in different way, you come up with different words. Same thing with proteins. Add the letters in different ways, you come up with a different protein structure and function. So we have the primary amino acid sequence, and scientists talk about that a lot. Then the secondary amino acid sequence is this spiral, so sort of the local neighborhood and the shape. It starts out cooked spaghetti, but then because of non-covalent interactions between the water and the amino acid chain, and also between the amino acids in the chain, we get some local neighborhood folding that's called secondary structure. And then the overall three-dimensional shape of a protein is called the tertiary structure. So when we say aquaporin looks like a donut, we're referring to its tertiary structure. Its structure gives it its function. We talked about that as one of the themes in the book. In Chapter 1, structure and function are connected. And then quaternary, some proteins function in as more than one protein in a complex. So here's just a protein that is functioned together by a group of small proteins. So we got these 4 different levels of proteins, and aquaporin is a great example to look into this stacking like that. Now we're going to start talking about this idea of making this, synthesizing this chain of amino acids. So here's just a drawing, and it shows this series of amino acids, and so here's an amino acid, and here's an amino acid joined together by this peptide bond. And this is this length here is referred to as sort of the backbone. So if you imagine that proteins are like cooked spaghetti, this is this strand that can wiggle and form the different shape chained through the length of the protein. And these are individual amino acids that are joined together in this chain. And it's a specific pattern. Here's the pattern shown here. And they get made one letter at a time, one amino acid at a time, rather. And so scientists just label a protein based on its primary amino acid sequence, label it based on when it gets added to the strand. So to synthesize aquaporin, we first add a glycine, then an alanine, then a serine. And I said you don't need to memorize these names or these structures, but here's something you should be able to identify, and that is each amino acid has a portion that is unique. So the backbone is common, and what are called side chains or R groups are unique. So there's the side chain for glycine. There's the side chain for alanine. And I said you don't need to memorize the names of the structures, but if you saw this amino acid chain, you should be able to state whether these amino acids are hydrophobic, water-fearing, or hydrophilic. And there's a nice table in your book that categorizes the side chains into, this is very hydrophobic because it's CH3 and carbon has the same electronegativity as hydrogen. So these are the kind of non-polar covalent bonds found in fats and oils that separate from water. So if you shake oil and water together and let it settle, the phases separate because the oil is hydrophobic, like that. So it's a very hydrophobic side chain. Then here's one that's hydrophilic, and it's hydrophilic because that OH is a polar bond. So it has some mixed characteristics. Here's another one that has some mixed characteristics. It's amphipathic. There's a big region here that is nonpolar. This benzene ring means that it's got 6 carbons with a series of hydrogens bonded to it. But it remains hydrophilic or polar because of that OH group, like that. Here's one that's hydrophobic. And so you should be able to identify that, and the book has a table with the amino acids organized based on which ones are charged. That would be hydrophilic and polar, blah, blah, like that. Here's just a drawing of the peptide bond being formed by a chemical reaction. So we have 2 separate amino acids, and they're going to be joined together here into a covalent bond. And what's being lost, if you drew this out, what's being lost is a hydrogen, 2 hydrogens, and an oxygen. So what's being lost is water. And there it is there. So in this chemical reaction, we go from 2 amino acids to 1 molecule of amino acids joined by a peptide bond plus water. This is a chemical reaction that's called a dehydration reaction. And that's what happens in your cells. Every one of your cells is doing this dehydration reaction to synthesize all the different proteins you need. The opposite reaction is happening in our stomach and small intestine, and that's called hydrolysis. So proteins being broken down by digestion. As I said, you know, we will eat proteins from another organism, and then we'll digest them down to individual amino acids for ourselves to make the proteins that we need. And so here's just some labeling of an individual amino acid. And just to show some features of it. And then this just shows that when the protein is being synthesized, we have amino acids being joined together, and a long chain of subunits, common subunits called a polymer, and chemistry. And so the dehydration reaction leads to the synthesis of a polypeptide, a protein polymer. And in chapter 5, the book points out that many many biological macromolecules are based on that polymer and that dehydration reaction. I just want to show this again from our first video in our course, video number 1. We were talking about insulin. We said that in some cases when you eat, there'll be a little insulin in the food you eat. So the protein that we talk about in our diet is actually literally proteins like insulin and a variety of other proteins present in the food we ate. And we digest it, and we break it down to individual amino acids, send those through the blood to cells that need to make proteins. All cells do, and the cells will chain the amino acids together to make a protein pattern. To do that, they follow the template. You have DNA. In the case of the insulin protein, it uses the insulin gene that includes a code calling for the specific amino acids that are going to be used. Remember, this concept is pretty important. And in the case of insulin, if we ate some hamburger that had insulin protein in it, that would be cow insulin, and our body's not going to use that. It's going to digest it, and it's going to use it and use the amino acids to synthesize human insulin. Same thing is true for aquaporin. Aquaporin is made in every plant and animal, so it doesn't matter whether you're a vegetarian or not. Every time you eat something, it almost certainly has some aquaporin protein in it, just a little bit of aquaporin protein. And we never use the aquaporin. We need it desperately in every one of our cells, but we never use it in our diet. Instead, we break the protein down to individual amino acids. The amino acids go through our blood, and we make human aquaporin and other human proteins. The book talks about a different example in introducing proteins, and we can look at, this is another introductory textbook. We can look at introductory textbooks. They're all introducing the same thing because they're really important concepts. And here are some proteins, and this is just showing very common that some people have sickle cell anemia. And they have a mutation in the DNA, and normally the DNA would code for the amino acid glutamic acid, and a mutation leads it to form a protein, hemoglobin protein. This is the primary amino acid sequence. This is normal, and this is mutant. Just has one change. And these are all normal amino acids, but they got the chain has one error in it, one difference in it. That's a normal valine and normal glutamic acid. It's just that glutamic acid is very hydrophilic. The R group or side chain is very hydrophilic, and valine is very hydrophobic. So some different chemical characteristics, like the qualities of chemistry that we learned about already in our class. And that leads to a slightly different shape in the hemoglobin protein, and we get a disease out of it because the protein's defective. We talked about that with aquaporin also. People can inherit a mutation in aquaporin. And just to scroll back to this, the mutation is here, and it leads to a change. Let's say we go from pink to blue. So the protein amino acids are all fine. We've just put the new and wrong amino acid in this place due to a change in the DNA sequence. So the DNA is where the mutation is. It leads to defective protein. Could be any protein. In this case, the insulin gene. There could also be more DNA, and it could be an aquaporin gene. Have a mutation, boom, it'll make a defective protein. All the amino acids are good, but we've changed one or more of the amino acids, and now it's defective. And here's just an image of that where we're looking down. Here's the donut, and we're looking down the donut hole, looking down the donut hole into the cell, and there was a mutation. And remember in the previous video I said that scientists just number amino acids in a chain, starting with the first one that's added. And at position 192, this mutation led from a change from the amino acid asparagine to the amino acid leucine. And it is physically now blocking the channel so that water cannot get through the channel. And so the function is defective. The purpose of the protein is let water in. The function is defective. So this is, again, example of one mutation that leads to a defect in the protein, leads to some sickness. Here are mutations that lead to errors in the structure and function of aquaporin. Again, we're looking down the hole of the donut. There's water. And here's a mouse that was inherited from its parents a defective version of an aquaporin gene. And as a result, it wasn't able to get water out of the cell rapidly enough to keep its eyes wet. And it got a kind of glaucoma during its life. And that can happen to a person too. So, remember back in an early video, I talked about this idea that here is normal Arabidopsis plant, this little weed, And here's a mutant Arabidopsis. Here's a mutant, and it doesn't let a mutant in aquaporin doesn't let water in fast enough. What would happen if we ate these two things? Would they affect us differently? The answer is no, because the issues with the DNA, and before we use DNA or proteins, we digest them down to individual building blocks and build what we want out of them. On the other hand, if the person has a mutation in an aquaporin, so they're unhappy, nothing we eat could help improve the situation. You know, you could eat aquaporin from a plant. Would that help? Nope. Because the person's going to break it down to the individual amino acids and then build the ones we want. This just shows that in addition to, you know, the significance of aquaporin in plants, there are lots of our cells that need to let water in and out really fast. And remember that we learned about this concept osmosis, and the function of aquaporin is to allow osmosis to occur faster. But it doesn't do anything else. It doesn't affect how water or other molecules move in or out of the cell. The cell might shrink or swell, and aquaporin doesn't affect it. It just allows those changes to occur quickly. Here's back to this primary amino acid sequence, just to review that again, because there's several important concepts there, and I think we covered them all already. Here's a concept that comes up in the book, and that is that really often we understand that there's a protein, and the protein could get its structure, three-dimensional structure, could get destroyed by harsh agents like these, and that would lead to a denaturation or an unfolding of this protein. So we go from a protein that looks kind of like a donut to one that looks just like cooked spaghetti. And what's being broken here are the non-covalent interactions. In the case of heat, remember how we talked about how temperature or heat is held in molecules by how much they vibrate. And we basically have vibrated this so much we shook it apart. The covalent bonds are still there. The heat did not affect them. Okay, all right, so next thing is, here are, and the book has this, and again, you can find this on the web. Here is the detailed structure of the atoms and the covalent and bonds and non-covalent bonds that lead to this kind of spirally structure in aquaporin that leads to about 8, I think, strands, spirals, that lead to a hole in the middle of a barrel. So we could get down to the individual amino acids to look at the very details of these structures, and that's something we just don't have to do. We don't have time to do in introductory courses like that. These are the interactions that would be disrupted when the molecule gets denatured. So there was a spiral, and the aquaporin was a series of spirals, and those get unfolded and looking like a piece of cooked spaghetti. There are several ways of non-covalent interactions that lead to a strengthening. Here's actually a covalent bond that leads to a strengthening of this, is the backbone of the amino acids joined together, like that. And these are some examples of some of the interactions between amino acid side chains that help strengthen or stabilize the three-dimensional structure. Like that. So it turns out that aquaporin actually has a quaternary structure. The aquaporin donuts get held together in groups of 4. And but we can talk about aquaporin tertiary structure because that explains what the function of the protein is. And the structure is donut-like. It's a channel. It lets water in and out. Very selective. And that's where we're going to end on this story about the protein structure of aquaporin.